The purpose of today's video is to introduce you to chromosome territories. So far, we've talked about chromatin structure in terms of its lowest level of organization. We've talked about the DNA being modified and the DNA being wrapped around histone proteins to form nucleosomes. We've also discussed the histones in the nucleosomes being post-translationally modified by, for example, methylation and acetylation. And we have examined what impact these modifications have on the interaction between the DNA and the histones. Lastly, we have spoken a little bit about how some of this packaging is more condensed and thus inaccessible to transcription, and some of it is less condensed and accessible. But so far, we have not looked at higher order packaging, or how exactly all those chromosomes, 46 for humans, actually fit into the nucleus. Here is the most simple model of how we can fit chromosomes into the nucleus, represented by this yarn ball with the yarns of different color representing different chromosomes. Everything is just randomly stuffed into the nucleus. As long as we can fit it in, we don't care how it's in there and what pieces of the various chromosomes are close to each other and possibly interacting with each other. In the right panel here, you see another representation of this model, which is called the spaghetti model. On the left panel, you have the alternative model, that of chromosome territories. This model basically proposes that chromosomes have a home address in the nucleus. They are found in a specific spot, they don't randomly intermingle, and they have neighbors, other chromosomes that are typically found close to them. So we have two very specific models, the chromosome territories model versus the spaghetti model. Now these two models make some very specific predictions. If we damage a specific region in the nucleus, the distribution of damage along the chromosomes would differ depending on the two models. In this figure here, the black dye corresponds to an area in the nucleus that we have damaged. In the real experiment, this was done with a laser. If we look at the left panel, which depicts the chromosome territory model, we can see that the damage is very localized. Only a few chromosomes are damaged, and there are many chromosomes that aren't damaged at all. In contrast, in the spaghetti model, we damaged many of the chromosomes. Here is this outcome represented in graphical form again. Either we have a few large areas of damage, or the damage is going to be smaller and along a lot of chromosomes. So A would be the territory model, B would be the spaghetti model. Now what did the actual experiment find? Here is the data from the actual experiment showing Chinese hamster cells that were damaged with a laser in just one region of the nucleus. And the damage was then revealed with a radioactive label, which shows up here as the little black dots. The damage is localized to just a few chromosomes, marked here by arrows, and most of the chromosomes remain intact and undamaged. This finding argues that chromosomes were actually organized in the interface nucleus into territories. With new technologies, scientists much later were able to get fluorescent images of chromosome territories, starting out by just labeling all the DNA from an individual chromosome, shown here in A. Using these techniques, we learned that some chromosomes tend to be at the nuclear periphery, while others tend to be in the interior of the nucleus. When researchers investigated the positioning of individual genes, there are two shown in red and green in panel B, they discovered that the localization differed from cell to cell. This is illustrated in D, where every dot is the location of a specific gene in a different cell. There's a lot of variability here, but the distribution is not random. This gene tends to be more often in the interior of the nucleus. Here is another example of chromosome territories with a more sophisticated labeling technique. Each of the chicken chromosomes in this image is labeled with a unique set of colors, seen in panel C. If you apply this mix of labels to a mitotic chromosome spread, you get panel B. Each chromosome is a different color. If you apply the labels to the interface nucleus, it reveals the chromosome territories in panel D. This experiment reveals that most of the chromosomes are near the nuclear periphery in this cell, and also that the two homologs of a single chromosome do not hang out together. They're in separate regions of the nucleus. So why is localization in the nucleus actually important? Additional research has shown that if we look at a cell where a specific gene is turned off, it resides at the nuclear periphery, shown in red in the left panel of B. If we now go ahead and turn this gene on, we can see that it has moved towards the center of the nucleus, on the right side in panel B. So the localization of a gene in relationship to the nuclear periphery versus the nuclear center provides information about its transcription status. Why are transcriptionally active genes at the nuclear center? It turns out there are regions in the nucleus that are enriched for all the various components of the transcription machinery. These regions are called transcription factories, and they contain genes from different parts of the chromosomes that all need to be transcribed. Only in the last 10 years or so have we been able to look at the 3D chromatin interactions in detail. This figure from your book summarizes the most often used techniques. They all begin by cross-linking, or gluing together pieces of DNA that are physically close to each other in the nucleus, irrespective of their location on the linear chromosome. Then, various molecular biology tricks are used that allow the researcher to retrieve these pieces of DNA and determine what pieces are close to each other. Please refer to your book for more details on these techniques. These high-resolution 3D investigations revealed that most chromatin interactions are local. Things that are close to each other on the linear DNA strand tend to interact with each other more often than things that are far apart from each other. There are exceptions to this general rule, and one of them is shown here. At the beta globin locus, when it is turned on or expressed, the upstream regions form two loops, allowing the enhancers to interact with the promoter of the appropriate globin gene. Interactions like these are seen as many enhancers and promoters. In summary, the nucleus is organized into chromosome territories. Nuclear localization correlates with transcription status. Silence genes tend to be at the periphery, expressed genes tend to be in the center. The probability of two sequences interacting decreases with distance, and enhancers generate loops to allow their interaction with promoters.